Hello, I'm Dr. Jackson. Welcome back to the Bio 122 video lectures. This is video number 11 on homeostasis and thermoregulation out of chapter 33, talking about animal body, basic form and function. So the first thing we're going to talk about is just the concept of homeostasis, which just simply means that organisms tend to maintain a steady state or a steady internal balance regardless of the external environment. So an example of that, the most common type of example we're going to talk about called negative feedback loops, you need there's certain components of a feedback loop that are necessary. First of all, you need to have some sort of stimulus. And an example of that would be body temperature. It could also be blood glucose levels. There's literally thousands of different stimuli in the body. But in order to initiate the process of homeostasis, you have to have some way of detecting what the level of that stimulus is. So that means you have to have some sort of sensor. And in general, that becomes the role of your nervous system. So neurons in the skin and carrying information to the brain will tell the brain how to regulate the different stimuli in the body. Because the next step is that once you know what the current condition of a variable is, such as body temperature, and let's say that it is too high. Instead of the normal body temperature of 37 degrees centigrade, say it's 40 degrees centigrade. So you have to have some way to change it. And to decide how to change it. So the control mechanism is generally going to be something found in the brain. There's going to be a temperature regulation center. In the brain, it's the hypothalamus. And then you have to have some way in which the brain can trigger a control of something. It's got to go to some sort of muscle in order to make a change. And that's called the effector, something to cause an effect. For controlling body temperature, an example of an effector would be the sweat glands that then can release water and allow water to carry away the excess heat in evaporative cooling. So that will hopefully return the body temperature back down to a normal body temperature. But there can be numerous different effectors that one control mechanism can be working on. And body temperature is a good way, is a good example of that, is that while you can sweat, we can also change the regulation of blood flow through the body, sending more blood to the skin so your skin gets flushed. And so there's multiple ways. It's not usually just one effector. So what the normal example that we just saw is what's called negative feedback. And well, if there's negative feedback, there's probably going to be an opposite of that. And sure enough, that's called positive feedback. But the vast majority of homeostasis is going to rely on negative feedback. And students sometimes get confused about that because they think of the word negative as being bad. But I want you to think of negative as being good, because this is something that will return a stimulus that is going beyond normal and would reduce the difference back to normal. So it's going to return it to a normal range. That's why we call it negative. It's a term from engineering. As opposed to positive feedback actually makes the difference larger and larger and larger. So it's actually the opposite of homeostasis. So you only see positive feedback in normal physiology in very specific circumstances. And I'll show you one of those examples today. So since the most common is negative feedback, let's start with that, because negative feedback loops will always counteract any change change in the internal environment. And remember, homeostasis is trying to maintain the same. So almost all biological systems use negative feedback to control things like temperature, glucose, your blood pH, the calcium in your blood. All those type of things are negative feedback systems. So let's look at one example in detail. A typical negative feedback loop that is regulating blood glucose levels. So this actually lets us bring in a couple of organ systems and also talk about the endocrine system. And in this example, there's two possible situations. Well, three possible situations. The first situation is normal, okay? So the normal or desired blood glucose level should be 100 milligrams of glucose per deciliter of blood. However, what if it exceeds that? So we can look at two other situations, a high blood glucose or a low blood glucose. Let's start with the high blood glucose level. So it's high because we have a sensor that's detecting the amount of glucose in the blood built in. It's a protein that's built into the pancreas, actually. And the sensor in the pancreas will detect that the actual blood glucose level is actually 120 milligrams per deciliter. Or remember, the set point or the desired blood glucose should be 100 milligrams per deciliter. So that means we have a difference of 20 milligram per deciliter, 120 minus 100. So what's going to happen? This one doesn't require the brain to do anything. In this case, the pancreas is capable of generating the response and determining which way that response should go. So there are two types of cells in the pancreas. One of them is called the beta cell, and the other one is called the alpha cell. We'll talk about the beta cells first because when they detect an increase in blood glucose levels, they start producing insulin, which is a protein. It's a hormone, but it's just simply a protein. So they create it and they release it into the blood supply. So the pancreas is the structure down here, the beta cells producing insulin, and it goes into the blood and it travels all through the body. And as it's doing that, the action of insulin is that it controls the ability of cells of the body, in particularly the muscle cells, it increases the rate at which the glucose transporter will move glucose from the blood to the inside of the cell. So it increases by a factor of 10 the rate at which glucose will be extracted from the blood and put inside of a cell. And as soon as glucose enters a cell, it undergoes glycolysis and then enters into the stages of cellular respiration to create ATP. It's really the only use of glucose. And so the cells will use as much as is given to them. So what we're doing with insulin is we're controlling how easy it is for glucose to get into the cells or to remain in the blood. So if you're putting more into the cells, you're reducing the amount of glucose in the blood. That's not the only thing insulin does. It also goes to the liver. And in the liver, it tells the liver cells to start taking glucose cells and combining them together into a new molecule known as glycogen. Glycogen is a storage form of glucose, but it's not glucose anymore. So again, both of these steps are removing blood glucose from the blood. So the end result is the blood glucose level will start decreasing as insulin gets released. So now we're going to have a revised actual glucose level. If we started this off at, say, 120 milligram per deciliter, we end up now with 110 milligram per deciliter. Well, the difference is now 10. 10 is still more than desired, but it's less than what it was before. So we were at 20, a difference of 20, now we're at a difference of 10. So if we continue releasing insulin, we continue this negative feedback loop until the actual equals the desired, at which point the difference will be 0. So this is why we call it a negative feedback loop. Each loop, we keep making the difference smaller and smaller and smaller. We're subtracting from it. So that's why it's called negative. Well, now what happens if we have the opposite situation where now you haven't eaten anything for several hours and our glucose level, instead of being 100 milligram per deciliter, drops down to 80 milligrams. So we have low blood glucose. We still have a difference of 20, but now it's a difference in the opposite direction. Pancreas is still going to be involved, but the pancreas, instead of the beta cells, it's going to be the alpha cells release a different hormone. Instead of releasing insulin, they're going to release glucagon. And of course, the beta cells have stopped releasing insulin. So now we're going to get a lot of glucagon in the blood. So that glucagon is going to travel to the liver. Just like the insulin traveled to the liver, the glucagon will go to the liver and tell the liver, hey, remember all that glucose that you stored as glycogen when insulin was telling you to do so? Glucagon reverses the signal so that the liver now takes glycogen and turns it back into glucose and adds it to the blood. The glucagon has no regulation of individual muscle cells because there's no glucose in those cells, because as soon as the glucose came in, it turned it into ATP. But without the insulin, we're going to slow down the speed at which glucose is transported into the muscle cells. So that's going to have the effect of increasing over time the amount of glucose that's in the blood. So now, when we had dropped down to 80 milligram per deciliter, now our revised blood level is 90. Instead of being a difference of 20, our difference is 10. So you see, we're doing the same thing. We're making the difference smaller. We're talking absolute values here. So don't go, don't think about either one as being positive or negative. They're both negative, okay? Because we're making the difference smaller and smaller and smaller until eventually, as the liver continues to convert glycogen back into glucose, we're going to come back up to the desired 100 milligram per deciliter. So a healthy pancreas is able to maintain a very stable level of blood glucose even when you're eating and when you're not eating. So throughout the day, you don't get much variation. The disease of diabetes prevents glucose from doing its job. And there's different types of diabetes, so it may be the pancreas is not actually doing its job, or the glucose is not correctly doing its job. And that will lead to wild spikes and highs and lows of blood glucose levels. Okay, so that's one of the most typical examples of negative feedback. So let's show an example of a positive feedback loop. And in this case, here is a common one for at least a portion of the population. And it doesn't happen very often, even in that portion, childbirth. The actual process of labor is a positive feedback system. So oxytocin is a hormone that's critical for the reduction of this behavior. So I'll just use the laser pointer here. So here we have a fetus at the moment of being born. So this is going into labor. And the fetus is within the uterus, which is a large container, stretched container of smooth muscle. So it's able to contract. So now the question is, what's the trigger for contraction And the smooth muscle contracts when it receives a signal from a hormone. That hormone is oxytocin. Oxytocin causes the muscles in the uterus to contract, and as they contract, they end up pushing the head of the fetus towards the cervix and the vagina for birth. So, where does the oxytocin come from? The oxytocin comes from the brain. So now we end up with a circle. So let's kind of just pick up at a random part on this circle. Let's start with the brain. So the brain is going to stimulate the pituitary gland, which hangs just beneath the brain, to release the hormone oxytocin. Oxytocin goes into the blood, travels through the circulatory system, and gets into the uterus. And when it gets there, it causes the muscles of the uterus to contract. When the muscles of the uterus contracts, it pushes the head of the baby towards the cervix, causing it to stretch. Buried within the tissue of the cervix are neurons that are stretch receptors. So they detect when the skin or the tissue of the uterus is being stretched at the cervix. The more they are stretched, they send signals to the brain to release more oxytocin. Okay, that's what the positive means. The action of oxytocin actually sets up the situation so that more oxytocin is going to be released. So now that more oxytocin is being released, the uterus contracts even more than it was before. The baby is pushed even stronger into the cervix. That stretches the cervix even more, which then causes the brain to release more oxytocin, which causes more contraction, which causes more push against the uterus, the cervix, and which causes more stretch of the cervix, on and on and on. So this is now you understand that process of labor is a series of contractions becoming stronger and stronger and stronger and stronger and stronger until they get strong enough to actually stretch the cervix. So this is what we're talking about when we're talking about when someone's in labor, how dilated are you? So that's how much stretch there is on the cervix, because the cervix has to stretch and stretch and stretch until it's large enough for this baby to pass through it. And so that is a rare example of a positive feedback system. Now note, this doesn't happen very often. It only happens to a small number of the population. It never happens to a male. And it only happens at brief periods of time in the life of females that are giving birth. But another feature of a positive feedback loop, it's used for a very specific reason, but it also has to have an off switch, because the nature of a negative feedback system is that it's always going to return you to a place of stability. So the stimulus that was creating the feedback in the first place is not there anymore because it's returning you back to normal. This isn't like that. A positive feedback system actually keeps getting stronger and stronger and stronger. So how do you ever stop the contractions? There has to be an off switch. And the off switch here is very simple. Stretching of the cervix is what's causing the brain to release oxytocin. But why does the cervix stretch? It's because the baby's being pushed into the cervix. So the off switch is birth. Because once there's no longer a baby in the uterus, you can contract the muscles in the uterus as much as you want, but if there's nothing to push, there's going to be no stretch of the cervix. So the cervix is going to go back to its original shape. That's going to stop the signal to the brain, and so oxytocin is going to die off. And that's what happens after childbirth, is that over a period of time, the contractions get less and less and less and less until they're no longer present. Okay, so there's only a couple other examples of normal positive feedback, and I'll actually cover those at different times in the semester. But this is the first one we'll talk about. Okay, so I just want to kind of finish up this section talking about another issue about homeostasis that sometimes gets confused. Is don't get confused that homeostasis and the word called adaptation. So two words here, acclimatization, so acclimatization. The climate is the environment. So this is something that can happen to an individual because of a change in the external environment. And this is something that happens over the period of weeks or months. And an example shown here is people climbing Mount Everest. You don't just jump right to the top of it. Climbing Mount Everest is actually a process that takes a few months. And one of the reasons is people will go to what's called base camps, and there's several base camps at different altitudes because the peak of Everest is so high, there's not enough oxygen to effectively support the human brain and the human muscles. So most people are actually wearing oxygen tanks when they do it. But in the meantime, they're also trying to to acclimate themselves to the low oxygen environment of the mountain. And so as you go to the base camp, initially you're very short of breath all the time. But over a period of a week or so, your blood cells actually start increasing in number. So you have greater oxygen carrying capability. You also start increasing the number of mitochondria, making it more efficient to produce ATP. But we become much more efficient at transporting oxygen. And so then they will pack up and they'll go another few thousand feet higher and then stop at another base camp and stay there for a week. And that will again increase the ability of the body to function well at a high altitude. So that's the process of acclimatization that can occur and change. We also see this happening during warm to cold environments, because if you go to a warm environment, we can actually start increasing the amount of salt that's in your sweat, or decreasing the amount of salt so that you don't become imbalanced as a lot of water is coming out. And vice versa, going into cold, you don't sweat as much. Another example would be typically for animals during the cold will grow thicker fur, and in summer will shed. If you own dogs or cats, you know that's true. And in some cases, animals even change the entire size of organs. Birds do this. Will change the size of their liver because that will increase the amount of heat they can produce using metabolism in a cold environment. So there's quite a few solutions, but this is not evolution. This is just changes over a period of time in an individual in response to the environment. So don't confuse that with evolution when we talk about adaptation, which is the evolutionary change that occurs not in an individual, but over multiple generations. So that's going to be based on the passage of alleles, mutations and genotypes in the passage of alleles, and changing the ability to survive in that environment. Okay, so just a short little note on those two different words that are often confused. Okay, so that's going to complete this section talking about homeostasis, and that's going to complete what we're going to talk about with this chapter. So this completes module 1. All right, take care.